Right, well, I hope everyone can see my screen, and I'd like to thank the organisers of this webinar series for the Middle East for inviting me to speak to you today. I shall be talking about the vitrification of oocytes and embryos, and we'll also be giving you an opportunity to participate in a sense, in that there'll be some polls and also some questions, and please get involved and answer these. They're completely anonymous, but it's just interesting to see what types of practices are going on out there. So firstly, I'll cover some of the key variables in vitrification that you may wish to optimize. Then I shall provide a detailed overview of the vitrification protocols for oocyte, cleavage stage embryo, and blastocyst cooling and warming. And then I shall consider the risk management and safety of vitrification and cryostorage. And finally, I'll summarize what's been covered, hopefully leaving ample time for any questions that you may have. So cryopreservation has many uses, primarily really the avoiding gonadotoxic cancer chemo or radiotherapy, so for fertility preservation, making donation more efficient, enabling delayed parenthood, and dealing with any unforeseen events that might impair treatment, and also it can make treatment more convenient for patients that may not necessarily be able to be present on the day of, let's say, oocyte retrieval. And blastocyst cryopreservation, of course, also allows time in which to receive PGT results and also can contribute to the cumulative live birth rates. So our first poll, so just curious to know, at what stages do you vitrify? You may vitrify at more than just one of these stages. So many people, it'll be oocytes and blastocysts, but we're just interested to know what stages you vitrify at. Okay, so as expected, most people are vitrifying oocytes and blastocysts, whether biopsied or not. But interestingly, some of you are also vitrifying at the molar stage and also at the zygote and day 2 cleavage stage. So it's interesting to know that there's quite a wide range of stages at which you're vitrifying in the Middle East. So when wishing to optimize vitrification, there are many variables that you need to consider. Perhaps the most significant one, of course, being gamete and embryo quality, which will be largely dictated by female age and culture conditions. Nevertheless, the other variables listed here may also have an impact upon lab and clinical outcomes following vitrification, and not least variations in operator skill, expertise, and consistency. Now, it's commonly assumed that the choice of media containing specific permeating and non-permeating cryoprotective agents is an important factor to consider. However, there's actually no clear evidence in favor of one or other of the cryoprotectants. And besides, different vitrification kits share similar composition, thereby lending themselves, in fact, to use in universal protocols. And it's more important, therefore, to optimize any given vitrification kit within your own lab. So you can see from these 6 different kits here, the ethylene glycol in combination with either DMSO or propanediol are the most common permeating cryoprotectants found within these kits. And despite some minor differences in pH buffers, supplements, and slight variations in protocols, all these vitrification kits can be optimized for individual lab settings. And of course, there are many cells and tissues still stored within cryobanks around the world that have been vitrified or slow frozen. And many clinics still receive samples from other centers that were cryopreserved using different kits and protocols. So in this case, the possibility of using a universal warming protocol can simplify the management of the warming procedure. And in 2014, Parmigiani and colleagues first looked at this, publishing a pilot study using human oocytes of a single universal warming protocol based on subsequent steps with 1 and 0.5 molar concentrations of extracellular cryoprotectant, irrespective of the freezing protocol, whether that be vitrification or slow freezing. And so they concluded that such an approach could actually simplify lab protocols, make staff training more straightforward, and also make it easier to manage the warming procedure. And they followed that up with this study where they looked at universal warming, testing it on 315 blastocysts. Each of these blastocysts was randomized at cooling and then again at warming to either Kitazato or Sage vitrification kits. And as you can see, the female mean age and survival rates were statistically comparable between the study groups. The blastocyst survival rate ranges from 98.4 to 99%, and implantation rates in different groups were also comparable, ranging from 18 to 22.5%. But to achieve competence in vitrification, it's necessary to be consistent in all steps of the procedure and carefully manage the main parameters that can affect your results. And in this respect, you need to bear in mind that protocols are usually designed for use at room temperature or at 37 degrees C. However, of course, the temperature will affect the speed of the process, and therefore the normal distribution of time for equilibration, for example, will be faster at 28 to 30 degrees C, let's say in a warm or pretty warm lab, than at 20 to 24 degrees C, let's say in a cooler or more typical range of room temperature. So first poll coming up, during equilibration, what volume of medium do you use? Remember, this is anonymous, so please join in. Okay, that's quite reassuring that most of you are using between 1 to 300 microliters, and then there's a bit of a range around that. The next most common is 50 to 99 microliters and 300 to 499 microliters, and then the slightly fewer are using less than 50 microliters. So nobody's actually using particularly large volumes at 500 microliters or more. So that's interesting. All right, so in this respect, the volumes of the cooling and warming media used are actually really important. So larger volumes may reduce the effect of errors. And you need to remember that if you're dealing with small volumes of media, the risk of increasing the osmolality due to evaporation is greater. And one of the big risks of vitrification is osmotic shock. And you can see in this in these graphs on the right-hand side, these are 15-minute equilibration periods at room temperature, and you can see there's a reduction of 25% in the media volume when just 20 microliters is used for equilibration versus only 1.2% when 500 microliters is using. So if you are using, and I'd say from this figure here, especially if you're using less than 300 microliters for equilibration, you need to be particularly careful about minimizing evaporation. So if you're starting out, it's actually probably better to start with large volumes until you achieve satisfactory results before making any changes. And of course, if you make changes, you should make them one at a time. It's also important to determine the optimal equilibration timings within different labs by observing the mean time taken for cell re-expansion to 90% or more of their original volume after placement in equilibration medium, because obviously the water is a smaller molecule, leaves the cells straight away, they shrink, and then it'll just take a bit longer for the permeating cryoprotectants to fill the cells. And once you've established a mean time for equilibration, you can then incorporate that into your standard operating procedure as your standard practice within your lab. And remember that the process of equilibration is optimized by moving the cells through different areas of the solution. Since vitrification solutions contain higher concentrations of permeating cryoprotectants plus disaccharides, they're more viscous than equilibration solutions. So unfortunately, the material will initially float to the surface, so you need to keep track of it and bring it back down to the bottom of the well. And but you also need to make sure, as you can see from these photomicrographs here, of re-expansion of an oocyte, 5 seconds, 30 seconds, 60 seconds. It's imperative that if the protocol says 60 seconds vitrification, that you allow the cells to remain in that vitrification solution for 60 seconds to make sure that you achieve complete permeation of cryoprotectants. But then you need to load straight away onto your carrier. And most importantly, as soon as you've loaded onto your carrier, you've got to go straight into the liquid nitrogen. So if it's less than 60 seconds, so be it, straight into the liquid nitrogen. You can't afford the risk of that very small volume that you've loaded into starting to evaporate. And again, you've got problems both with osmotic shock and toxicity. So what about carrier loading volumes? They're inversely correlated with cooling and warming rates, and therefore the tendency is to use the smallest loading volumes that you can safely use. Desired volumes can be achieved using an adjustable pipetta, such as the stripper. I particularly like to use a device like that because then the loading volume is consistent every time. Alternatively, you can pipette out known volumes onto your carrier at a known magnification onto your dissecting microscope and just get an eye for what is the right volume. So you probably want to be aiming for somewhere in the range of 0.1 to 0.5 microliters. And if you pipette out too much medium, you can always aspirate the excess away, leaving a thin film surrounding the material that you're trying to vitrify. So let's start with oocyte vitrification. These can be simple two-step procedures, such as the Embryo procedure there, and they can be sequential steps, again, to mitigate the risk of osmotic shock. But again, it's even better if you, probably with an oocyte, a larger cell in the body, if you use a more gentle procedure involving more than just two steps to slowly increase the concentration of the equilibration medium. Again, different volumes may be used, but larger volumes are more likely to minimize evaporation of media, particularly during equilibration, which can last up to 15 minutes. And certain dish designs can help you handle the material. And I've always found that the use of 2 drops of vitrification solution is particularly useful. So what do you do? This is just a yes, don't know, or no. For oocytes, do you use the more gradual sequential addition of equilibrations medium? Okay, so yeah, the vast majority, nearly 70% of you, do indeed use that more gentle approach. That doesn't surprise me. Okay, so now I'm just going to show you a short video showing how to go about using that gentle procedure to cool oocytes using the Sage vitrification kit. As with any precisely timed processes, it's really important that you get everything ready. This procedure is performed completely at room temperature. So you want to place your oocytes into a 20 microliter drop of MOPS or Hebes buffer and leave them there for 1 minute, taking them away from the light source to maintain that room temperature. And then the next step is to make sure you've mixed your pre-room temperature equilibration solution and add 20 microliters to that 20 microliters of your buffer to the oocytes. So, and then equilibrate for 3 minutes. And then follow that up with another 20 microliters. So just gradually, slowly increasing the concentration of the equilibration solution surrounding the oocytes. So another 20 microliters there. And again, leaving them at room temperature for another 3 minutes. And then finally, getting up to the full concentration of equilibration solution required by adding 240 microliters to that same drop. And then leaving them to equilibrate for a further 9 minutes. So that's a 15-minute equilibration in total. And while those oocytes are equilibrating, you can prepare your carrier, which is properly labeled and has been witnessed to confirm that that labeling matches the labeling on the dish. And at this stage, you pipette out your 2 150 microliter volumes of vitrification solution. So you don't want these vitrification solutions evaporating during that equilibration period. So remember, these are more viscous and maybe more prone to evaporation. Always replacing the lid in between times. And then transferring your oocytes at the end of the equilibration period to that first well containing vitrification solution, getting those oocytes under control onto the bottom of that well and leave them there for 30 seconds. So then when you transfer them to the second well of vitrification solution, the oocytes are exactly where you want them. You don't have to mess around with them. You can now get your carrier ready. And at the end of that second 30-second period, you can load your oocytes onto the carrier and immediately into liquid nitrogen. And then make sure you fill the cap with liquid nitrogen, and then the black mark helps you find the way into the cap, and then slight clockwise twist tightens the cap, and that's done. Now for embryos or blastocysts, most people use a simple two-step protocol, and again, volumes typically range from 100 microliters to 1 mil. Again, the same principle applies, that larger volumes and certain dishes may help. The other thing that may help with blastocysts is collapse of the blastocele. So you can see from this study there are various methods to collapse the blastocele and to remove the fluid from the blastocele cavity, so that when you come to equilibrate the blastocyst, you can do so in a much shorter period of time, because you're not having to allow for the permeating cryoprotectants to diffuse across a fluid-filled cavity. Generally, laser collapse of the blastocele is considered the safest and quickest method. The other thing to bear in mind is loading of blastocysts onto a single carrier. So ideally, in an ideal world, we'd always load a single oocyte or embryo onto each carrier. But in practice, it's not unusual for example, 1 to 5 oocytes or 2 to 3 cleavage stage embryos in particular to be loaded onto a single carrier. However, this study demonstrates the potential risk inherent in overloading a carrier, since although you can see in the upper table that there was no significant difference in cryosurvival rates when 1 or 2 blastocysts were loaded, in the lower table you can see that fully intact blastocyst cryosurvival rates were significantly lower, however. Okay, another video just to show you that simple 2-step procedure which you can use for your cleavage stage embryos and your blastocysts. Again, get everything ready. You don't want to stop halfway through because you've forgotten something. And again, because it's the stage vitrification kit, it's being performed at every step at room temperature. These vials would have been allowed to reach room temperature for about 20 to 30 minutes before use, after taking out of the fridge, mixed. And then you can, because these volumes here are quite large, you can pipette out both your equilibration solution and your vitrification solution. And also with cleavage stage embryos, you're tending to equilibrate for a shorter period of time, and certainly with collapsed blastocysts. So you transfer your embryo or embryos to your equilibration solution and equilibrate. So typical ranges are 5 to 7 minutes for cleavage stage embryos, 5 minutes for collapsed blastocysts, and 12 minutes for expanded blastocysts. Again, during the equilibration period, you can prepare your carrier. Again, very important to get that witnessed as being correct labeling for that patient. And now transfer to your vitrification solution. So here you've got to get that embryo or the blastocysts under control on the bottom of that dish before you can aspirate them into the pipetta. And at the end of that 60 seconds, you can then load the carrier, again with a minimal volume. If it's a hatch blastocyst, maybe allow a bit more volume, and then straight into the liquid nitrogen. Again, make sure you fill that cap with liquid nitrogen before capping the device. Okay. So this brings to mind, there are different types of carriers you can use, and there are different types of storage conditions, vapor or liquid phase. So practitioners choose either open or closed carriers, either because of the ease of handling open carriers with their known high cooling and warming rates, or because of a wish to minimize the risk of cross-contamination by potential pathogens within liquid nitrogen. But you can see in this study, rates of recovery, cryosurvival, blastocyst formation, and DNA damage within blastomeres was compared between the cryo loop and open carrier and HSV and cryotip closed carriers, and also between storage and liquid nitrogen and vapor storage. And there was no significant difference between any of them. So really, it's up to you to decide what you would prefer to use. So in terms of your cryo storage, do you have a monitoring and alarm system in your laboratory for your cryo storage devices? So yes, no, or you don't know, you're not sure. Excellent. So 3/4 of you do, which is really good. Those that don't, I would suggest you think seriously about it. I've had a liquid nitrogen storage, liquid nitrogen storage dewar fail. I mean, luckily not one that I had any samples in, but I did learn from that the liquid nitrogen level goes down extremely quickly once that the core develops a crack, and the liquid nitrogen can get through the core and evaporates very quickly on the outside of the metal container. So following initial warming, you then need to undergo further dilution of the permeating cryoprotectants and rehydration of the cells. And that's achieved by managing the osmotic gradient using diminishing concentrations of non-permeating disaccharides. Now, one thing that's really important to appreciate is that, yes, most efforts are aimed at achieving a very high cooling rate using open carriers. So, some publications have often included some discussion of the possible contribution of the warming rate, but there's actually been only very few experimental attempts to separate the importance of each step. And this is one such example. This study by Secchi and Mazur showed the importance of the warming rate using the mouse oocyte model. And in their experiments, you can see, particularly when presented with the cryosurvival rate against the warming rate in that graph on the right-hand side, you can see essentially there's a linear relationship between the warming rate and cryosurvival, almost independent of the cooling rate. So it's the warming rate that's really critical. And when it comes to warming protocols, they're generally applied to oocytes, cleavage stage embryos, and blastocysts. Though you can see that the dilution and washing following the first warming step, which is performed at 37 degrees, of course, that can be the dilution and washing can be performed either at room temperature, as with the Embryo kit there, or at 37 degrees, as with the SAGE kit. Regardless, the key to success is ensuring that the temperature of that first warming medium is maintained at 37 degrees. So you get that really rapid warming rate, and that it's performed for that full minute before the next step, because you need to get those high concentrations of cryoprotectants, permeating cryoprotectants, out of the cells as quickly as possible. So last video, just to show you how warming is achieved, again using the SAGE vitrification kit. So in this instance, you can do everything at 37 degrees. You don't have to mess around using ambiplates or stages at different temperatures. Really important, have everything absolutely ready. And most important, have your warming media capped and your warming dish in an incubator at 37 degrees for at least 20 to 30 minutes. You need to validate this in your own hands to make sure that they're both at 37 degrees when you take them out of the incubator. Now, here in preparing the dishes, you prepare your dilution and your washing media first. So 500 microliters of 0.5 molar sucrose warming solution goes into your dilution and washing dish. And 250 microliter volumes of mop solution, in this case with this kit, into successive wells for your washing steps. Now, it's deliberately done this way because the last thing you want to do is to remove your warming media and your warming dish out of the incubator. So there's minimum time for those to warm up. So here you can see dispensing a minimum of 500 microliters of the 1 molar sucrose warming solution into your preferred dish. I tend to prefer using a center well organ culture dish. Once your heat is staged, which hopefully you've calibrated to maintain that warming medium at 37 degrees, then double checking, witnessing it's the right embryo, and then quickly into your warming solution. In most instances, the material will just drop off the carrier. Sometimes you need to tap the carrier, and you can see this full 1 minute of warming in that warming solution. And then you can transfer your oocyte or your cleavage stage embryo or your blastocyst to the 0.5 molar dilution solution, and you leave it in there for 3 minutes at 37 degrees. And then you can transfer to your first washing solution and leave it in there at 37 degrees for 5 minutes. And then into your second washing solution, your mop solution, leaving for a further 5 minutes at 37 degrees C. Again, putting the lid back on the dish each time. And then finally, you can transfer your oocyte or your oocytes or your cleavage stage embryos or your blastocysts to your culture dish. And then you can put that culture dish into your incubator, ready for ICSI, further development, or embryo transfer. Okay, so we need to remember that vitrification is not without risk. And you can see from this data collected by the European Commission that serious adverse events in reproductive medicine occur primarily during processing and testing, followed by storage. And these are mainly due to human error and tissue or cell defects, equipment failure, etc. So these serious adverse events usually occur because there's been an error or break in the chain of custody. Therefore, to manage and mitigate such risks, it's absolutely vital to correctly label all consumables used to handle material during vitrification and cryostorage with the patient's name, their date of birth, and the date of vitrification, and a unique identifier, such as the patient's medical record number, and to witness each and every step of the procedure, especially including the cryostorage location. You don't want to be stuck with trying to find what's been cryopreserved when you come to have to do the warming. Electronic witnessing using either barcodes or radiofrequency identification tags is considered the best way in which to mitigate risk and avoid human error. A combination of both RFID tags and barcodes is actually required during the vitrification and cryostorage process, since RFID tags have yet to be perfected for use in liquid nitrogen. And recently, digital cryostorage and billing programs such as Embryo Options have been introduced to help clinics administer and track their patients' embryos while in cryostorage and when transported between IVF centers. Again, such a system ensures informed consent for cryostorage, disposal, or donation to other patients or to research. And it's a great system, for example, for auditing your cryostorage tanks. So, come to the crunch question. Do you think that vitrification is safe? Yes, you don't know, you're not sure, or you disagree. that is safe. You think it's unsafe. Okay, well, that's quite reassuring. So most of you, over 90%, agree that vitrification is safe. So that's good to know you're confident in what you're doing. And the data was tend to support what you think. So, of course, an important consideration is the safety of oocyte and embryo vitrification and the various risk factors associated with it, such as handling and processing of these material in such small volumes, and their ability to generate healthy pregnancies, births, and neonatal outcomes, including, of course, any long-term effects on babies. And we also have to bear in mind, although there are no documented cases of this, published reports of this, there's also the potential risk of microbial cross-contamination. Reassuringly, if we look at this study, looking at vitrified and non-vitrified control oocytes, they observed equivalent aneuploidy rates among both arrested embryos and usable blastocysts derived from those oocytes, and those that were transferred maintained equivalent reproductive potential. In this study, there were 26 paired transfers, one blastocyst originating from a control oocyte and one from a vitrified oocyte, and the ongoing pregnancy rate per embryo transferred was similar, with 53.9% from vitrified oocytes versus 57.7% from the control oocytes. And also global DNA hydroxymethylation patterns analyzed by immunostaining day 3 embryos, again derived from fresh or vitrified oocytes, are found to be similar, and at least half of the zygotes obtained after ICSI of those oocytes were euploid, regardless of whether they were fresh or vitrified. And they also did time-lapse analysis, which showed that there was no significant difference in cleavage timings, predictive morphokinetic time intervals, nor average cell numbers between the embryos that have developed from those oocytes. Also reassuring is several studies reporting comparable obstetric and neonatal outcomes and comparable development and health of 2-year-olds following pregnancies resulting from the use of autologous vitrified oocytes and fresh oocytes. So in summary, how are you going to start doing vitrification or continue doing vitrification successfully? Of course, select only good quality oocytes and embryos. Be really fastidious with your control of timings, temperature, osmolality, and your pipetting. You don't want to contaminate one solution with a more concentrated solution, for example. Evaluate the time for equilibration, because that will differ between labs and between different stages of development. Load in minimal volumes, so you get those fast cooling and warming rates. Be really careful about how you handle the carriers, especially if you're going to transport them between labs, and especially if you're going to do audits of your tanks. And as I think I showed you, the warming step is critical and reveals the sum of all prior steps, of which each may be detrimental if mistakes occur. Some selected bibliography there. If you need any of these publications, then don't be afraid to ask. Okay, I think I can maybe stop sharing there, and happy to handle any questions that you may have. I notice there are some in the Q&A.
Yeah, thank you, Steve. We have a few questions.
Okay.
The first one, would warming on 37 degrees be better than room temperature?
You mean dilution and washing? Okay.
Yeah.
Well, I'm going to have to share my screen again for that one. Can you allow me to share my screen?
Okay.
Okay. Now that's a really interesting question, because literally in the last weeks, the first publication I've seen on this has been published. So let me just see if I can. Oops, maybe I shared the wrong one here. Oh no, here we go. So, whoops.
Yeah, you stopped sharing.
Sorry, let me try again. There we go. So this has just been published very recently. In fact, when I first looked at this, it was still under peer review, but it's now been reviewed and published. And it's a very interesting study because it's quite a large study. So, albeit a retrospective study, they looked at 1,734 FET cycles. And these embryos were either cleavage stage embryos or blastocysts, and they were vitrified using a Kitazato vitrification kit according to the instructions for use for both cooling and warming. Of course, all embryos were warmed at 37 degrees C initially, and then were either diluted and washed at either room temperature or at 37 degrees. So you can see the first column here is everything done at 37 degrees. The second column, the first column is warming at 37 degrees and subsequent dilution and washing at room temperature. And the second column along is everything at 37 degrees. And what they found was they controlled for baseline characteristics between the two groups. They matched them 1-to-1 to try and avoid confounding. And what they found, and you can see this with those p-values, they found that the clinical pregnancy rate and the implantation rate of blastocyst transfers was significantly higher when they did all the warming and the dilution and the washing at 37 degrees C. However, interestingly, when they looked at the clinical pregnancy rate and implantation rates of day 3 transfers, although they were, again, higher in the all 37 degrees C group, they weren't significantly higher. Maybe there were insufficient numbers or whatever. But kind of contrary to what I guess we tended to understand with slow freezing, for example. We always used to do dilution and washing after slow freezing at room temperature because we were always afraid of those larger cells lysing if the re-entry or the rehydration of the cells was too rapid. But it seems that study at least suggests that in fact there may be an advantage to doing the dilution and the washing at 37 degrees C. But of course, this can vary from kit to kit. So again, I'd love to see more studies of this type undertaken because, like I said, that's the only one I could find where they controlled for the kit. They controlled, of course, for the same incubators, same dishes. The only difference was that they were diluting and washing either at 37 degrees or at room temperature. So should I go to the Q&As or are you going to give me the next one, Hadil?
Yeah, we have plenty actually.
Okay, good. I like questions.
What's the main thing to take into consideration when choosing the carrier?
What's the main
thing to take into consideration
when choosing carriers?
When choosing the carrier.
Yeah, I think like all things in life, whatever you find easiest to handle. So that involves, of course, if you're still writing by hand, I used to find it pretty amazing that my younger colleagues could read and write tiny writing on the carriers. Using, of course, labels makes life a lot easier. You can pretty much use any carrier for that. But I think ultimately, whatever you find easiest to handle. As I said, there's no published report of any cross-contamination in liquid nitrogen of pathogens from one patient's sample to another. So I don't think there's any paranoia over using a closed carrier. So if you prefer using an open carrier and you can handle that more easily, which is probably more likely to be the case, then I think that's perfectly acceptable. You have to be pragmatic about this, and the most important thing is that you manage to do these procedures in an efficient and timely manner, and that often comes down to what carrier you feel comfortable using.
Yeah. All right. The second question: what day is preferable to freeze? Is it day 3 or 5?
What is the best to vitrify, at day 3 or day 5? Okay, well, that's again a really interesting question. I will admit that in the past we used to vitrify both day 3 and day 5, and there were several reasons for that. One was that, of course, if you had, prepared for let's say a transfer on day 3, and your day 3 embryos, let's say, didn't survive, you could also actually warm a day 5 embryo and transfer it early into the uterus. So the embryo will wait for the uterus to become receptive, and so if it was a blastocyst, it can just sit there and wait for the lining of the uterus to become receptive. The other thing that I particularly like about vitrifying on day 3 is that you can actually validate the viability of that vitrified and warmed embryo, because you can keep it in culture to day 5, and it can prove its viability by being able to develop to a blastocyst by day 5. And then you've got a really viable blastocyst to transfer. It's survived vitrification and warming. It's proven its viability because it carried on developing. You've got a really high chance of achieving a pregnancy with that. With blastocysts, we can vitrify and we warm, but other than re-expansion of the blastocele, we don't have a real handle on whether that blastocyst is viable or not. Of course, if there's cell lysis in the blastocyst, then that's not such a good sign. But if everything else looks fine, again, you've only got re-expansion of the blastocele to tell you that those cells are actually viable and pumping fluid into the blastocele cavity. So it's another reason why I particularly like the idea of vitrifying on day 3. So yeah, it's a really difficult one to answer. I think if you vitrify and transfer on day 3, you will not probably get as good results as if vitrifying and transferring on day 5. But if you vitrify on day 3 and culture to day 5, my feeling is you're going to get as good, if not better, results than vitrifying on day 5.
All right. We have other question about getting some lysis in cells of blastocyst after warming, and what would be the reason for that?
Okay, well, that's always a hard one to answer, because, of course, when you come to do the warming, as I said in the summary, it's the sum of everything else that you've done. So, for example, you may get cell lysis because of toxicity, osmotic shock, and that could happen both during cooling and during warming. So it's very hard. It could, of course, simply reflect the viability or the quality of that blastocyst that you decided to vitrify. So we know, for example, that published data on this showing that better quality blastocysts suffer less cell lysis and are more viable, produce higher pregnancy rates and lower miscarriage rates than blastocysts of not such good quality. So it's difficult. So I think if you are experiencing that, then my advice would be to start at the beginning and do a risk assessment of exactly how you're going about selecting the material that you choose to vitrify, how you're going about doing the cooling, preparing your dishes, doing your cooling, your equilibration and your vitrification, how you go about loading your carrier, immersing in liquid nitrogen, and how you go about ensuring that your warming is definitely at 37 degrees in that warming medium, and that you're doing everything according to the IFUs in terms of timings. So it's just a matter of reviewing everything you're doing to rule out, if you like, operator error. So again, there's no easy answer to that one, unfortunately, or no single answer to that one. It's a complex answer.
Yeah, it is. I think we can take only one last question, right, Amir?
Go ahead, take 2 or 3, no problem. We have 4 questions left, so we can answer these.
I'll answer quickly.
Yeah, because we have plenty and we are running out of time, I believe. Do you recommend oil overlay for vitrification dish to reduce evaporation?
Yeah, you really can't use it for your vitrification dish. But you can use it for your dilution and washing dish. So because these permeable cryoprotectants, they can get into oil. You don't want to use an oil overlay on your vitrification dish. And also it's going to make it a bit messy when you come to pipette onto your carrier. But once you've done your warming, and you can then transfer that warmed material into your dilution medium and then on into your washing media, then by all means, if you want to use an oil overlay, because it's just disaccharides, they won't merge with the oil. They're not hydrophilic, so you can safely use an oil overlay if you wish on those steps. Next one.
What are your thoughts on rebiopsy and revitrification? Any recommendations?
On rebiopsy and revitrification? Okay, so as I said earlier on, vitrification is not without risk. And so if you're going to have to do something twice, you're doubling the risk. But if you have to rebiopsy, certainly make sure you've always verified that you've tubed your biopsy before vitrifying your blastocyst, or if you're doing biopsy at earlier stages, whichever stage you're working with, don't have to warm and rebiopsy because of that. But if you have to rebiopsy because you've got a indifferent result coming back from your genetics provider, then you don't have much choice. You just have to do it. But bear in mind that the results will not be quite as good as when you biopsy and vitrify once.
All right, and we have a question about for how long do we have to wait before the FET after warming?
No particular time. So again, it's up to you. If you want to, for example, if you've vitrified oocytes, then you probably want to be in timing with that time from when the ACG trigger was given. Once that oocyte has been reinitiated in terms of its maturation, the clock is ticking. You don't want that oocyte to become post-mature, so you need to do your ICSI relatively soon after warming. So just bear in mind the clock. And if it's a blastocyst, then again, you may wish to keep in culture for up to, let's say, 2 hours to see if you can observe re-expansion of the blastocele. And otherwise, if you're using some fancy transfer medium containing hyaluronan, then just follow the IFU for that. And make sure you don't have the embryo in that medium for too long before transfer. In the real world, we're usually at the behest of the clinician that needs to do the embryo transfer, but let them know, of course, let the patient know when they've got embryos that are viable and can be transferred. I think that was the last question.
Yeah, I believe so.
Anyway, if there are any other questions, then always feel free to email me with any other questions you may have.
Thank you, everyone, for joining us today. You will receive a short survey, anonymous as usual. It will take only 2 minutes to answer. We're looking forward to receive your answers, and we'll be sending the certificates of attendance to your email, and looking forward to welcome you to our next webinar. Have a good day. Thanks for joining.
Thanks for joining.
Thank you all.